Insulin, HPV vaccines, and some food additives are proteins widely used in society. In fact, they are in such high demand that they are mass-produced as recombinant proteins. Recombinant proteins are the products of recombinant DNA, which is a DNA molecule joined from at least two different sources. Recombination is used because the protein of interest is usually eukaryotic and are not easily obtained or amplified. Bacteria serve as factories for this purpose because they are easy to grow, multiply rapidly, can be manipulated with plasmids, and conveniently isolated. Recall that plasmids are used as cloning vectors because they are joinable with the gene of interest, transformable, and replicable. For the purpose of recombinant proteins, plasmids are used as expression vectors. The expression vector is different from a cloning vector because in addition to replication, it must achieve transcription and translation. Like a regular cloning vector, expression plasmids contain the selectable marker, such as the antibiotic resistance gene that allows for screening and selection of transformed bacteria, the ORI for replication of the plasmid, and the multiple cloning site, which allows the gene of interest to be inserted into the plasmid through restriction enzymes. Expression plasmids also contain a bacterial promoter upstream of the gene insert for transcription by RNA polymerase, a transcriptional terminator sequence, as well as a ribosomal binding site to translate mRNA protein. For recombinant proteins, one must control the extent of transcription, so that the bacteria produce a lot of the gene product only when instructed to do so. Quite often, strong and inducible promoters are used in expression vectors. These are based on the lac operon of E. coli. When lactose is not present, a repressor molecule binds an operator sequence upstream of the promoter, preventing RNA polymerase from transcribing the genes. Lactose, when present, is an inducer and releases the repressor from the operator. This allows RNA polymerase to transcribe the genes needed to metabolize lactose. The balance between inducer and repressor molecules determines the rate of expression, and this inducible system is applied to expression vectors. Let's review the recombination process with insulin, a eukaryotic protein implicated in diabetes. To obtain insulin in high quantities, the gene is first isolated, reverse transcribed to cDNA, and amplified through PCR. Second, the insert and plasmid are digested with restriction enzymes. Third, the gene is inserted into the expression plasmid. Fourth, the plasmid is transformed and replicated by bacteria on antibiotic spread agar. Lastly, bacterial machinery transcribes and translates the insulin gene in the presence of inducer molecules, such as methanol. The protein product can then be purified through appropriate tagging and chromatography techniques, such as ones that we will go over in this course.